everyone, today we are going to present the class project called DNMT isoforms. To begin with, we must remember the term epigenetics, which refers to the study of change in gene function that are durable and cannot be attributed to change in the DNA sequence. Such changes will affect the accessibility of DNA for regulatory and transcription factors without affecting the basic nucleotide sequence. Genetic machinery regulates gene expression by DNA methylation, histone post-translational modifications, and no-coding RNAs. Here, we will focus only on DNA methylation. The third question posed is functional characterization of DNMT isoforms and their role in methylation in humans, and the proposed objectives are functionally characterize the isoforms of DNMTs. To establish the role of DNMT's family, isoforms in methylation and expression regulation in humans. Identify cell lines and cell types with increased expression of DNMTs. And finally, to establish the interaction between DNMT's isoforms. As stated above, we will focus on DNA methylation, which is a vital process during development, cell differentiation, and tissue homeostasis. DNA methylation is a process in which the methyl group is covalently attached to the C5 of the cytosine residue and is catalyzed by evolutionarily conserved DNA methyltransferase isoforms, as can be seen in the image. The methylation process takes place in the CpG islands of the genome, which are guanine and cytosine-rich, phosphate-bound regions of DNA. In mammals, there are 5 DNMTs, of which 3 are catalytically active: DNMT1, 3A, and 3B, while 3L and 2 are not. Therefore, in this study, we will focus on those that are catalytically active, or also called canonical. The isoforms will be responsible for one type of methylation. As we can see, DNMT 3A and 3B are responsible for de novo methylation. In this case, we can see an embryonic cell, of which we have its DNA wrapped in histones. DNMT 3A and 3B will allow the embryonic cell to differentiate into a cell type. This will become a skin cell. In addition, these DNMTs are essential for the establishment of global DNA methylation parents and show high expression in embryonic tissues. We will have DNMT1, which is responsible for the maintenance of DNA methylation after the differentiation. As we have already seen, methylation will occur in the CpG islands that are found throughout the genome except in the promoter CpG island. Promoter regions are regions of DNA that contain regulatory elements that control gene transcription. The DNMT1 is also involved in the maintenance of methylation parents within the genome after cell division. The 3 canonic DMTs are located in the core of the cell. DMTs consist of a regulatory domain, N-terminal, and a catalytic domain containing the conserved domains between DMTs. The N-terminal differs in size between DMTs and is responsible for interaction with DNA and other proteins. The domain present at the C-terminal is called SAM. DMTs obtain the methyl group from SAM and transfer it to cytosine. The BAH1 and BAH2 domain appears to act as a protein-protein interaction module specialized in gene silencing. The function of this domain related to chromatin binding. PWWP domain is a functional domain of transcription factors that regulate developmental processes. It may also be involved in protein-protein interactions. On the other hand, ADT domains are considered to regulate protein, DNA, and protein-protein interactions. In this slide, we can see the isoforms of each of the DNMTs. DNMT1 has 3 isoforms. The first 2 have the same domains as DNMT1, and the third does not have the DNMT1 binding domain. DNMT3A also has three isoforms. The first two share the same domains and the third has no domains. And finally, DNMT3B has 8 isoforms, of which the first 4 share the same domains as DNMT3B. To evaluate the expression of DNMTs, three factors, cell line, developmental stage, and cell type, were analyzed. For the first factor, cell lines related to different types of cancer were observed for all three DNMTs. In addition, high expression was observed in early developmental stage 4, 5, and 6. And finally, in cell type, it is observed that the most expressed cell types are immune cells, such as T cells and endothelial tissue cells. Alteration in the de novo and maintenance methylation processes can lead to the development of different diseases. As you can see, one of the most common is cancer. Normal adult cells, most CpG sites are methylated, except for promoter CpG island. Promoter regions are regions in DNA that contain regulatory elements that control gene transcription. In the image on the right, we have a normal methylated adult cell. But are we missing something? Yes, the TET, called human 10-1 translocation, which is in charge of demethylating the DNA and maintaining a balance so that everything runs smoothly. Now, if we have an imbalance due to hypermethylation or demethylation, problems arise. In the image on the left, there is evidence of a hypermethylation process that can cause cancer pathologies in many cases, such as acute myeloid leukemia in the bone marrow cell line. On the other hand, it has been shown that a low expression of DNMT3B leads to hypermethylation and the expression of the immunodeficiency syndrome 1. After our last uptick, we did a review in INTAC where we found that our DNMTs can interact together when there is not a correct methylation process, and this results in cancer in the early stage of development. On the other hand, it was evidenced that DNMT3A and 3B maintained an interaction in de novo methylation. The isoforms that report interactions in INTAC are presented below. An example of this is the DNMT1 isoforms that interacts with the G.7 protein responsible for regulating the stability of multiple proteins involved in various cellular processes, including DNA damage response, transcription, and epigenetic control of gene expression. In conclusion, the role of DNMT3A and 3B in de novo methylation and DNMT1 in maintenance methylation was identified. On the other hand, the domains present in the isoforms allow us to obtain an understanding of their functionality. There is a high expression of DNMTs in the early stage of development, and the cell types with the highest expressions are T cells and endothelial tissue cells. Actions allow us to observe that DNMTs can interact with each other. Also, that there is a little information about isoforms interactions. Therefore, the study of these isoforms is necessary to have a better understanding of the biological processes in which they are involved.